Hello students, welcome back to my channel Competitive Botany. In this video, I am going to give you a detailed lecture on crossing over. So before going to see the crossing over, you need to know chromosomal theory of linkage was proposed by Morgan. He proposed that a phenomenon is nothing but the the genes present on single chromosome, they do not tend to separate. They goes as it is to the they went as it is to the next generation. They do not separate during gametogenesis. They are going to the next generation as it is without change. They goes to the next generation as it is without change. This is called linkage. So the genes present on single chromosome, they do not tend to separate during gamete formation. They as it is inherited to the next generation without change in their genes sequences. Okay, so this phenomenon is called as linkage. So occasionally this is called complete linkage. Occasionally they are separated by a phenomenon called as crossing over. Crossing over is nothing but the mutual exchange of genes between non-sister chromatids of homologous chromosome pair. So, in any organism, you can find pairs of chromosomes. These two are the homologous chromosomes. We are getting one from mother and other from father. During the formation of during fertilization, so we are getting one from mother, one from father. Okay, so these two chromatids are called as sister chromatids belonging to one chromosome, whereas these two chromatids are called as sister chromatids belonging to one chromosome. Okay, so here I am telling mutual exchange of genes in between non-homologous chromatids of non-sister chromatids of homologous chromosome pair. So you can imagine this is first chromatid, second chromatid, and third chromatid, fourth chromatid. So for easy understanding I am telling. So these are the chromatids. These two are sister and these two are sister. One chromatid is non-sister to 3 chromatid. One is non-sister to 4 chromatid. Okay. So in the same way, second chromatid is non-sister to third chromatid, as well as second chromatid is non-sister to fourth chromatid. So here I am telling mutual exchange of chromatin segment in between non-sister chromatids of homologous chromosome pairs. These two are non-sister chromatids. They overlap one another. Wherever they overlap one another, they are showing a chiasma-like structure where they are going to exchange their chromatin material. So after exchange of chromatin in between 2 and 3, we are going to get new combination of genes. Whatever the genes are present over here, they come to here. Whatever the gene content present over here, they come to here after exchange of chromatin in between them. Then we are getting new combinations of genes. So due to this, variations observed, variations are reported in progeny. Now I am going to give you the definition for crossing over. So the exchange of chromosomal segments between non-sister chromatids of homologous chromosome pair. Okay, homologous chromosomes. So the exchange of chromatin in between non-sister, exchange of chromatin occurs in between non-sister chromatids of two homologous chromosome pair. Okay, so exchange occur in between 2 and 3 or 1 and 4 or 3 and 4. They also the non-sister chromatids. Okay, wherever the overlapping of chromosomes occurs, there you will find the crossing over phenomenon. So this is called, this cross-shaped configuration is called as chiasmata. This cross-shaped configuration is called as chiasmata. So let's see the frequency of crossing over. Frequency of crossing over. So frequency of crossing over occurs between two genes is expressed in terms of distance or percentage of crossing over. So in general, percentage of crossing over is directly proportional to the distance between genes. That means, so these are the two genes and these are the another two genes. So here you can observe the less distance. Here you can observe more distance. This gene is far to this one. So here the crossing over is less and here the crossing over percentage is higher. Why? Because they are directly proportional to each other. If the distance is high, then linkage is less. That's why they tend to separate from each other. If the distance is less, so the linkage is high. That's why they do not, there is a phenomenon of linkage, they do not tend to separate. Their crossing or less chances for crossing over and chiasma formation. Okay, so that means the percentage of crossing over between genes represents the distance between two genes. Okay, it has a great significance. So you can calculate the frequency of crossing over is equal to number of recombinants present or produced in the test grass by the total number of progeny into 100. Okay, so this is the equation. You can calculate the frequency of crossing over. Okay, so I will explain you in detail. So these are the homologous chromosomes. For the easy understanding, I draw like this, red color and blue color. So you can observe two homologous chromosomes. So wherever the chiasma formation occurs in between two homologous chromosomes, so of non-sister chromatids of two homologous chromosomes, there the crossing over occurs. okay, resulting in new recombination chromatids, new recombined chromatids. They are getting variations in their gene content. That means you can see whatever the gene content present over here is transferred to here. Whatever the gene content present over here is transferred to here. Okay, you can see the new chromatid formation. So this is called variation. So after during meiosis, it occurs, crossing over occurs in Pachytene stage of meiosis 1. So then after meiosis completion, 4 chromatids are going to 4 different gametes. This one is parental chromatid, as it is went to goes to the one gamete. This is the new recombinant is going to other gamete. This is also new recombinant is going to other gamete. This is again parental combination is going to other gamete. So here you can observe 4 gametes. 4 different gametes are produced. Among them 2 are recombinantal gametes. Remaining totally 4 gametes are produced from these chromosomal segments. Okay, so now you can see, so now you can calculate the frequency of crossing over. So for example, I am telling, okay, so now you can see the frequency of crossing over. How many recombinants are produced in the progeny? 2. How many total progeny? 4. So into 100. Okay, so you can calculate this. So percentage of crossing over is 50%. So this is the answer. Okay, in this way you can calculate the frequency of crossing over. So Hutchinson observed crossing over in maize plant. So he has taken, this is the best example for the crossing over. Okay, he has taken 2 types of seeds, colored full grains. They are crossed with colorless shrunken grains. So we are getting, as usual, gametes. Same gametes are getting from the parents. So they are combined to each other in F1. You are going to get the dominant individual, dominant hybrid, colored full hybrid is produced when it is test crossed with a recessive parent. So crossing over occurs in between these 2 chromatids. So when what will happen? So new combination of gametes will be produced. As a result, we are going to get new combinations in the progeny. These 2 are the new combination of new combinations, new varieties are produced in the progeny. Totally here also we are getting 4 progeny. Among them 2 are new combinations. This is the parent and this is also the parent. Among them 2 are recombinants and 2 are parental combinations. Okay, so you can see here colored full 4032 plants are produced, colored shrunken 149 produced, colorless full 152 produced, colorless shrunken 4035 plants are produced. Among them these 2 are the crossover products, these 2 are the recombinantal products only produced in 4%, 2% again 2%. These are produced in 48% and again 48%. Okay, so you should remember that recombinants are produced through a phenomenon called crossing over only. Okay, let's see the mechanism of crossing over. Let's see the mechanism of crossing over. So crossing over is the exchange of chromatin segments in between non-sister chromatids of homologous chromosomes. You know that. So in the mechanism, so for your convenience, it is divided into 4 subheadings. Let's see. First one, synapsis or bivalent formation. Synapsis or bivalent formation. Okay. During zygote substage of prophase 1 of meiosis 1, chromosomes are lie side by side. Homologous chromosomes are lie side by side. So due to they are paired due to mutual attraction between them. So resulting pair of homologous chromosomes is called as bivalence. This pairing is called as bivalence. Okay. So this phenomenon is also called as synapsis. Okay. So let's see the second heading, that is tetrad formation and chiasmata formation. So in Pakitine stage of prophase 1, so bivalent splits longitudinally. To form two sister chromatids of homologous chromosomes. To form two sister chromatids for each chromosome. Okay. So thus now bivalent consists four chromatids. Okay. So this is actually stage called as tetrad stage. Okay, now the homologous chromosomes twisted around each other. At the point of contact between non-sister chromatids, these two are non-sister, one is belonging to other chromosome, other is belonging to other chromatid. So at the point of contact of non-sister chromatids, so they are showing a cross-shaped configuration. So that is nothing but chiasmata. That is called chiasmata, cross-shaped configuration. At the chiasmata, the chromatids are going to break by the activity of an enzyme endonuclease. They are called as molecular scissors. Okay, then the broken segments of one chromatid is fused to the other, and the broken segment of other chromatid is fused to the other. So this fusion brought about by an enzyme linkage. This fusion is brought about by an enzyme linkage. Okay, the broken segment of one chromatid is fused with the other chromatid and vice versa. So this fragment is fused to this and this is fused to this one. Then new combination of genes is produced. Okay. Let's see the third heading, that is crossing over or exchange of chromatin material. So this is the third one. So you know that this exchange of chromatin segments occurs between non-sister chromatids of two homologous chromosomes. So the crossing over therefore includes breaking of chromatids and transport and also fusion. So after crossing over, so they repel each other. So due to attraction during zygote, they come together and paired each other. After exchange of their chromatin in between the two non-sister chromatids, the attraction becomes repulsion. They repel each other as the synapse attraction between them decreases. After synapses, after exchange of two chromatids, they repel each other. Then they are going to separate in the next session. Let's see the fourth heading. The fourth heading is terminalization. Okay, during diplotin stage, desynapsis begins and two homologous chromosomes in a bivalent are pulled away from each other. Okay, two homologous chromosomes, they pulled away each other. So the chiasma, present in between two homologous chromosomes, so it moves a zipper like manner towards the ends. Okay, so from here to here it moves a zipper like manner, so towards the end of the tetrad. Okay, this movement of chiasma towards the ends is called as terminalization. So at the same time, chromatids are shortened in diakinesis. Homologous chromosomes separate except at their ends. Generally, each event of crossing over produces two recombinant chromatids. They are called as crossover chromatids. For each cycle, each homologous chromosomal pair produce two crossover products as well as two non-crossover products. So in this way, crossover chromatids will have new combination of genes and the gametes carrying them will produce recombinant phenotypes. They are called as crossover products. Okay, non-crossover chromatids will contain parental combinations. They contain parental gene combinations. They are carrying them will give rise to parental genotypes. They are called as non-crossover products. Therefore, frequency of crossing over between two genes can be estimated by the recombinant progeny produced in the test cross. So the frequency of crossing over is equal to recombinants produced in the test cross by total progeny into 100. In this way, you can calculate the frequency of crossing over. Okay? So, let's see the theories explaining the crossing over. So many theories have put forwarded to explain the mechanism of crossing over. Among them, we are going to see some important hypotheses. So, first one is Johansson's hypothesis, proposed by the scientist Johansson in the year 1909. So he proposed that prior to crossing over of homologous chromosomes, so during zygote stage, they become paired. During pachytene, they become tetrad. During diplotin, at the point of contact, that means chiasma, the chromosomes break and rejoin. Due to that mutual exchange of chromatin in between two homologous chromosomes, new combination of chromatids are produced according to Johansson. He proposed that one chiasmata is to one crossing over. This hypothesis, he proposed this. Okay, so later another scientist came to know. He proposed another hypothesis. Second hypothesis is the torsion hypothesis. So this was proposed by the scientist Darlington in the year 1935. So according to this theory, two homologous chromosomes, they wrapped around each other. So each chromosome has one chromatid. Two homologous chromosomes, they wrap around each other. Okay, they wrapped around each other. Each chromosome has one chromatid. When new chromatids are formed, the dyad becomes tetrad. Then the dyad becomes tetrad during the wrapping only, when they are in wrapping condition only. Okay, so it becomes tetrad. Now the paired equilibrium. So after becoming tetrad, the paired equilibrium and longitudinal cohesion will become upset. When they become tetrad, the cohesion will become upset. So now the attraction between homologous chromosomes, that is replaced by repulsion. The attraction now becomes repulsion, which forces them to separate. Okay, so as the chromatids begins to separate, their coil unwinds in one direction and sister chromatids unwind in the opposite direction. They unwind in different directions. Okay, so now it creates a torsion between chromatids that leads to breakup of non-sister chromatids. So this hypothesis is nothing but torsion hypothesis. This torsion hypothesis is not much supported by many cytologists because the chromosomes are duplicated before the division stage. Okay. Let's see the next theory, breakage and exchange theory. So this was proposed by these two scientists, Stern and Houghton, in the year 1969. So they discovered the presence of two enzymes such as endonucleases, molecular scissors and ligase. Okay, so firstly endonuclease will break down non-sister chromatids of homologous chromosomes, and then ligase it governs the reunion of broken non-sister chromatids. However, there's a lot of controversy about mechanism of breakage and exchange theory. So let's see the next theory, contact first theory. This was proposed by a scientist, Cerebrowski. So according to him, two non-sister chromatids first contact and then cross each other, and then breakage occurs at point of contact of chromatids. The broken segments rejoin to form new combinations. So first the chromatids contact first, and after contact they are going to break, and after breaking they rejoin each other, resulting in new combinations. So this is the actual theory proposed by Cerebrowski. Let's see the next theory. Let's see the next theory, that is breakage first theory. So this was proposed by the scientist Muller. So according to him, chromosomes first break into two or more segments. Chromosomes first break into two or more segments. So the broken segments of non-sister chromatids. The broken segments of non-sister chromatids, they rejoin with an exchange of forming chiasma. They rejoin by an exchange of chromatin with the help of a chiasma formation. So this is the breakage first theory proposed by the scientist Muller. Let's see the next theory, duplication theory. This theory is also called as the copy of choice theory. This was proposed by the scientist John Belling. He visualized bead-like structures. They are genes. They are interconnected by non-genetic linking elements. So according to him, copying of one chromosome up to certain distance, is carried by one chromatid, and then it is switching over to other chromatid. So remaining distance is copied by other non-sister chromatid. In the same way, another chromatid also produced by another non-sister chromatid. After copying of half of this chromosome, it is switched over to this one. Now another segment is copied by this non-sister chromatid. That means he assumed that daughter chromatid is derived due to copying of one chromosome up to certain distance, and it is switching over to the other for the remaining segment preparation, remaining segment copying. Okay, so thus the new chromatids carry information from both the parents, both the parental strands, both the parental chromatids. This results in combination of new varieties. Let's see the types of crossing over. This is single crossover, double crossover, multiple crossover. So double crossing or single crossing over in this type, chiasmata occur at one point. Double crossing over, chiasmata occurs 2 points. Triple crossing over or multiple crossing over, in many places chiasmata formation occurs. Okay, we are going to see the factors affecting the frequency of crossing over. Let's see the first one, distance. So if the distance between two genes is more, then crossing over frequency is increases. If the distance is less, then it is leading to linkage. Crossing over frequency is decreases. Second one is temperature. So as the temperature increases, the crossing over increases. Third one, age. As the age increases, linkage intensity increases, so crossing over is decreases. So next one, interference. Interference is nothing but crossing over or one crossing over that affects the crossing over effect of the other crossing over. That means if you observe one crossing over in between two non-sister chromatids, it doesn't allows the other crossing over actually. So that means it doesn't allows the another crossing over in that chromosome. Okay, so this is called interference. So there is a less chances for the second crossing over actually. Okay, so let's see the next one. X-rays. Increases the frequency of crossing over. They increases the frequency of crossing over. Next one, gene mutations. They may affect the frequency of crossing over. Some mutations are known to decrease the frequency of crossing over. Let's see the next one. Chiasma formation. At one point, chiasma formation in one chromatid, so that decreases the chiasma formation near to this. Okay, one chiasma is decreases the chance of formation of another chiasma. This phenomenon is called as interference. So next one, centromere. So centromere tends to suppress recombination actually. Okay, so that means near centromere the crossing over chances are very less. Next one, inversions of chromosome. So inversions of chromosome segments that suppresses the chance of crossing over actually. So let's see, now we are going to see the significance of crossing over. So this is a universal phenomenon, has great significance in genetics. It provides evidence for linear arrangement of genes in chromosomes. Okay, it provides due to this, depending upon this, we can construct the chromosome maps. You can know the sequence of chromosomes. You can also construct the chromosome maps. Okay, so as a result, so new combinations will be produced through crossing over only. As a result, crossing over is play a role in evolution. It plays an important role in evolution.